My pleasure to introduce today's IVB seminar speaker, Bill Murphy from Wisconsin. Bill did his graduate work with David Mooney and then moved on to work with Neil Mercich at University of Chicago. And he's been at Wisconsin for 5 years now. Okay. I met him when I gave a talk at Wisconsin, I guess about a year and a half ago, and it's clear that he's got some very, very cool things going on in his lab with responsive gels and delivery motifs. He also just barely overlapped with a former student of mine in Milan Mercich's group who took over, I guess, part of his research, and I'm not sure if he made a mess of that or not. But so there's definitely some philosophical connectivities as well. Today he's going to be talking about smart biomaterials inspired by nature's mechanisms.
Okay, so thank you, Andrew, and it's a great pleasure to be here to talk to you a bit about what we've been doing in the biomaterials area. Much of what we do is inspired by how nature regulates cell behavior and regulates release of molecules in the cell's local environment. What I'll talk about today are a couple of examples where we're using nature as a guide and starting to move toward really understanding and regulating cell behavior using materials. So, as you all know, natural systems are built largely upon non-covalent interactions. So non-covalent interactions are ubiquitous in nature and are quite important in controlling cell behavior, controlling regulation of soluble and insoluble signals. Examples range from complementary DNA interactions to receptor ligand binding and really everything in between. So what we've been interested in is, based on clear understandings of intermolecular interactions in nature, trying to use some of these mechanisms to build materials that we think are intelligent, so trying to mimic nature's mechanisms. And each of the types of intermolecular interaction or sort of binding interaction that you see up here, we've worked toward leveraging to control signaling in the cell's local environment. And there are references indicated here if you're interested in a particular area. What I'd like to talk about today really are just a couple of examples to give you a flavor for what we try to do, and then ultimately I'll talk about some of the applications, directions that we're heading in. So first off, I'd like to talk about what we're doing in the area of intramolecular protein-protein interactions, essentially allosteric changes in protein folding as a mechanism to control material properties. So this work falls broadly in the category of dynamic hydrogels or dynamic materials, which are also often referred to as stimulus-responsive hydrogels. So the concept here is that dependent on the properties of the solution environment that a hydrogel sits in, it might be more or less swollen, it might have differences in mechanical properties, physical properties, and ultimately might result in changes in, for example, biomolecule release. Traditional stimulus-responsive materials have responded to physical-chemical inputs like temperature, changes in pH, changes in ionic strength, changes in the external electric or magnetic field or the presence of light. And really the canonical examples in the literature are materials that are made from polymers like polynipam, polyanisopropyl acrylic acid, acrylamide, that's temperature sensitive, polymethyl acrylic acid-based hydrogels that are pH sensitive, and electric field and light sensitive materials like polythiophene, but there are a broad range of examples in this case. So these materials are quite interesting from a physical chemistry perspective, but also potentially quite important in engineering applications. So there are dynamic materials that have been used as interfaces with biology to sort of control biological systems or to attempt to control biological systems. A few examples, so drug delivery systems, Nick Pepys, Alan Hoffman, and others have really pioneered the development of drug release systems that are based on these kinds of dynamic hydrogels. So the hydrogel shifts its properties, resulting in changes in release rates of a drug. Demonstration here from a paper published in 1996 in Journal of Controlled Release from Nick Pepys' group on streptokinase delivery. Cell culture technologies, so Teruo Okano's group and others have been developing cell sheet technologies, for example. So cells can be cultured on the surface of a temperature responsive hydrogel upon a temperature shift, the wettability of the surface shifts, the physical properties of the surface shifts, and therefore the layer of cells can peel up, and you then have a cell sheet that can be used in tissue engineering applications. That's another example of the potential for these kinds of approaches. And then soft actuators. This is one that gets described in just about every paper that's been published in the dynamic materials area. The potential for using these kinds of hydrogels as artificial muscle materials. Here's an example of a bending hydrogel developed in Mitsuru Akashi's group at Osaka University. This is based on polynipam. And then adaptive optics, like adaptive lenses that are also based on polynipam and polymethyl acrylic acid-based materials. So suffice it to say, there are lots of potential technological applications of these materials. One of the limitations potentially in the field, in terms of traditional materials, is that they respond to physical-chemical inputs like pH and temperature that can be not highly specific. So in some cases like drug delivery, one might want to develop a system that responds to some sort of specific biomarker and results in release of an agent that can affect a disease state, for example. And similar issues with cell culture technologies and soft actuators. So if one wants to get to sort of the next stage in development of these kinds of dynamic materials, where we can think about theranostics and other sort of emerging phenomena regulating biological environments, then these sorts of new concepts might demand a higher level of specificity. So this is the sort of thing that we and others in the field have been interested in, what we might call bioresponsive materials. So one of the broad sort of paradigm concepts in bioresponsive materials is leveraging natural functions. So proteins are the main mediators of dynamic natural functions in nature. And there are a few different classes of protein function that can be exploited to build bioresponsive materials. One is biocatalysis, so there are a variety of enzymes that are used that in vivo catalyze chemical reactions. There are ligand binding interactions that can be used and competitive ligand binding interactions that can be used to change material properties. And then there's conformational flexibility. In the area of biocatalysis, Takashi Miyata's group, Nick Peppis's group, Alan Hoffman's group, and others have developed a variety of materials that can change their properties in response to changes in biocatalysis. So the glucose oxidase-based hydrogels that are common in the literature now, but really pioneering when initially developed, are a good example of this. So glucose oxidase catalyzes a chemical reaction, resulting in a change in pH that then in turn changes the properties of a hydrogel network, resulting in changes in insulin release. So for type 1 diabetes, this is a potentially very useful technology. Ligand binding has also been used to develop dynamic hydrogels, bioresponsive hydrogels. This is a schematic from a paper published in Nature in 1999 from Takashi Miyata's group. The concept here is that a hydrogel contains as part of its backbone an antibody and an antigen. That antibody and antigen, under initial circumstances here, are bound to form non-covalent crosslinks in a network, so the cross-linking density is initially quite high. If one then flows in soluble antigen to compete with these interactions, the soluble antigen binds the antibody, dissociating the antigen-antibody complex, and you get increases in swelling, which can be useful for drug release and other sorts of technological applications. What we've been particularly interested in is this third really poorly explored phenomenon in the materials area, and that is conformational flexibility. So this is what I'll talk about today, sort of an alternative approach that's based on the ability of natural molecules to undergo complex conformational changes. So proteins undergo an array of complex conformational shifts in response to a broad range of inputs, including both physical chemical inputs and specific ligand binding. A couple of examples shown here that are very common and that I'm sure most are quite familiar with are hinge motion in the protein kinesin and hinge motion in the protein myosin. So these are two motor proteins that undergo this hinge motion. So we hypothesized early on that a molecule that can undergo this kind of hinge motion, if built into a three-dimensional hydrogel network as a functional component, essentially along the backbone of the gel, would result in a molecular change from an extended conformation to a collapsed conformation that could then be translated into a macroscopic change in material properties, essentially collapse of a full-scale material. So this is the concept that we started with. One of the things we like about this concept is the flexibility. So proteins can clearly be engineered to tailor their responsiveness via recombinant DNA technologies. And there are several hundred protein conformational changes that are well characterized. So there's a large set of building blocks that could be used for this kind of an approach. The protein that we started with is a protein molecule called Calmodulin. This is Calmodulin in its extended shape, what's often called a dumbbell-shaped conformation with a long internal helix, here shown in green. When Calmodulin binds to specific biochemical ligands in the presence of calcium, this is what happens. So you get this collapse, and you're seeing the cycling of this collapse here. So this is a tremendous collapse in protein structure that's actually pretty rare, at least in currently understood conformational changes, this magnitude of change. So we thought this would be a great candidate to start working with in our systems. So the building blocks we started with in these hydrogel systems were an engineered version of Calmodulin that contains cysteine residues on the two polar ends of the protein, so at the 34 and 110 amino acid positions of Calmodulin. These are shown in red here. And a polyethylene glycol diacrylate molecule. So the concept here is that the sulfhydryl groups on the end of cysteine can react with acrylate groups via Michael-type addition reaction that then results in a covalent linkage. And then once purified, this would result in the presence of a protein that's linked on both ends to PEG acrylate and therefore can be photo-cross-linked into a three-dimensional network. The cysteine to cysteine distance in the extended conformation of these engineered proteins is about 5 nanometers, and when collapsed, gets as small as about 1 nanometer. So this is a big linear collapse, and then once incorporated into a three-dimensional network, this is sort of what we hypothesized. Extended, we'd have a hydrogel with a relatively large mesh size, and then when exposed to ligands that bind specifically to Calmodulin and induce its conformational shift, like trifluoperazine or TFP, we would get this kind of collapse in network structure. So initially, does the reaction go? So we ran the reaction between the engineered Calmodulin and the PEG diacrylate with excess acrylate. And if we use excess PEG diacrylate, we get this reaction to go what appears near completion. If we then purify this out on a column, we end up with this molecule. So a Calmodulin unit with PEG acrylate on each end. And if we then expose this molecule to UV radiation in the presence of a photoinitiator, I-2959, we end up with a three-dimensional network. What you're seeing on the lower left here is a gel that was formed and then stained for protein content. And what you're seeing is the top view of a cylindrical hydrogel that's in solution. So you can see that there's no label that's diffusing out over time. The gel has got protein linked within it, and the protein isn't diffusing out. So if we quantify the incorporation efficiency of Calmodulin into these gels, we get somewhere in the neighborhood of 90% incorporation, which we thought of as quite positive. And we can vary the total amount of Calmodulin present in the gel by essentially mixing the Calmodulin PEG conjugates with just PEG diacrylate units. So this potentially provides a mechanism to tailor the responsiveness of the material. So the next question, once incorporated into a hydrogel, do these molecules actually undergo the conformational shift? Do the materials undergo a volume shift? And here again, you're looking at top views of cylindrical hydrogels, initially in calcium-containing buffer, then in the presence of trifluoperazine ligand that induces the Calmodulin shift. And you see this pronounced collapse in the volume of these materials. And then back in calcium buffer for recovery. So you can actually see that as the ligand diffuses out, we get recovery of the initial volume of these hydrogels. And as we vary the amount of Calmodulin included into the gels, we can vary the degree of volume shift of the materials. So for engineering purposes, this is an important result. So we get as much as about a threefold decrease in volume on the high end, which for bioresponsive materials is pretty positive. So we're enthusiastic about this. The binding stoichiometry is close to what you see in solution for Calmodulin binding to trifluoperazine, which is roughly 4 to 1 in solution, but it's not quite as high. So the suggestion here is that not all of the protein that's incorporated remains able to bind its ligand, but quite a lot of it does. This collapse in volume can be cycled. So what you're looking at here are multiple cycles of exposure to trifluoperazine ligand And so the material gets back to its initial volume and then can collapse again. And then we can spatially control this as well. So if we create a polyethylene glycol hydrogel and then create a Calmodulin-based hydrogel adjacent to it, but sort of in the same network, if we add the trifluoperazine ligand, you can see that the Calmodulin side collapses and the PEG side remains intact. So it's possible then to sort of spatially control things as a bioactuator. So there are a few different applications that I want to mention of these materials, and I'll focus on sort of the delivery angle because I think that's the interest of a lot of the audience, and obviously Georgia Tech is quite a leader in the area. So in terms of actuation, we've been able to develop materials by varying the local concentration of Calmodulin in a gel, by applying electric field, etc. Materials that can undergo anisotropic shifts in volume. So materials that bend, like this sort of hydrogel. Materials that collapse along an axis, like this kind of a hydrogel. And then what's potentially important for actuation is materials that have dynamic units that can do work on non-dynamic units. So that's what you're seeing here, but will become clearer in a movie. So this is the beginning of the movie. The interior of this hydrogel contains the dynamic Calmodulin-based component. The exterior is just polyethylene glycol static. And what you'll see as the movie progresses is this interior portion is collapsing and actually pulling on the PEG component on the outside and essentially doing work, curling it up over time. This is a time-lapse movie. So you can see that interior essentially pulling up the outside of the gel. So this is an early demonstration that one portion of a hydrogel might be able to do work on another portion of the hydrogel in sort of bioactuation or soft robotics applications. Another application we're interested in is triggering protein drug release. Here we've actually generated microspheres composed of these kinds of protein-based networks. So using approaches that Andrew Lyon and others have really perfected, we've been trying to make hydrogel microspheres that are then dynamic. So they look like this, essentially. The extended network looks like this, and then when ligand gets added, the collapsed network looks like this. And here, the concept is to be able to control protein release from a network. And there are multiple mechanisms for this, so I want to spend just a couple of minutes describing what these mechanisms are. So one concept is that in a network that has a low mesh size, the protein release rate is relatively slow. As the mesh size is increased, so in networks that have a large mesh size, then release of a protein drug essentially increases in rate, and the total amount of protein drug release increases in rate. So a variety of studies from Nick Peppis's group and others have demonstrated that if one generates a dynamic network that's initially collapsed and then undergoes a network rearrangement so that it's expanded, you can increase the release rate of a protein drug. What we were interested in is actually kind of an alternative. So this is an example. The streptokinase release rate can be increased by changing temperature and pH in a temperature-responsive delivery system. We were interested in a bit of an alternative phenomenon. So the other thing that's been borne out in the literature a bit from Dave Mooney's group and others is that if you apply a mechanical strain to a hydrogel that contains a protein drug, then it's possible to perturb that network so that protein essentially gets released. And this has to do with changes in interaction between the protein drug and the background network, as well as changes in convective flow of fluid in the network. So you're essentially squeezing out protein via mechanical forces. So our thought was that it might be possible to do this without applying external pressure, but instead essentially creating a network that undergoes a shift in conformation, shift in properties that results in changes in internal pressure of the network and squeezes out a protein drug. So we generated polyethylene glycol Calmodulin microspheres using an emulsion photopolymerization approach. This is what the microspheres look like in solution. So this is an oil and water emulsion where we do the polymerization and we purify out the components at the end of the process. If we, again, do our staining for protein at the end of the process, you can see that there's fluorescent staining in this spot in the protein-based networks. In polyethylene glycol hydrogels that contain no protein, this is where the hydrogels would be in a fluorescent micrograph, and you don't see any staining because there's no protein in the network. So the protein is incorporated stably into the network. And if we pass these microspheres through a syringe, we don't see significant differences in their diameter or in the number of microspheres. So this is potentially promising for applications of these materials. They're robust enough that they don't essentially fall apart when they go through a syringe. This is sort of a preliminary analysis of whether they might be useful. But then the big question is whether or not these particles will undergo the same kind of shifts that we've seen with the bulk hydrogels I described earlier. And they do. So if you look at distributions of particles before application of trifluoperazine and then after application of trifluoperazine here in gray, what you see is about a 50% decrease in volume of these microspheres, and this can be tailored based on the amount of calmodulin in the network. If you look at quantitatively the volume shift in response to a variety of different environments, you can see that in the absence of ligand, this blue curve here, there's essentially no change in volume of these hydrogel microspheres over time. In the presence of arginine, which is a positively charged molecule that's similar in charge to trifluoperazine but different in terms of its ability to interact with calmodulin, so it's a nice control, we see a flat line again, no significant decrease. And then importantly, in the presence of serum, we see no decrease in volume of these materials. So this is important, suggests that the interaction, the collapse of calmodulin and that of the network is biospecific. It's not happening in heterogeneous solutions. If we had two different ligands that bind to calmodulin, one trifluoperazine and the other chlorpromazine, we see significant decreases in volume down to about a 5-fold decrease in volume of these hydrogel microspheres. So quite a substantial shift. And this is a movie that shows what the shift looks like. What you're seeing here are microspheres floating in solution, in suspension. And at time 0, we're going to add a ligand that induces this shift in properties. If it'll play. Well, that's too bad. I'll play it at the end because I have it on my desktop, but I don't want to interrupt. But basically, in the first 30 seconds after adding the ligand, we see a substantial collapse of these microspheres. They change their optical properties as well as their volume, so potentially useful for delivery applications. And this again can be cycled, so we can drop the volume in response to trifluoperazine. The materials recover. We can drop it again; they recover. So this is a cyclable phenomenon. So our question was this sort of new concept. Can we translate a protein conformational change in a hydrogel network into a triggered protein drug release phenomenon? So can we trigger release at a specified time? So the concept is that this blue drug, which in this case is vascular endothelial growth factor, a factor that influences angiogenesis, can it be incorporated into a network, and then essentially can we trigger drug release in the presence of a calmodulin ligand? So if we look at the comparison between VEGF release from these microspheres in the presence of no ligand compared to protein release in the presence of the trifluoperazine ligand, you can see that when TFP is added at time 0, there's a substantial change in the release rate. And then at subsequent times, if we keep adding TFP again and again and again, we do see additional bumps in the release curve, but that become less substantial over time, largely because we're depleting the protein from the network. So the initial release is quite a lot of the protein that's incorporated. But the really important mechanism is shown here. So the possibility that we might be able to trigger release at a later time. So if we take a network that's releasing low levels of background VEGF over time, and then we hit it with trifluoperazine at 24 hours, you can see this large increase in release rate, and the amount of protein released is similar to what we see at time 0. So this is a demonstration that we can trigger protein release at a specified time, which for regulation of angiogenic processes could be quite useful, and that's sort of where we're going with some of the initial work. So this is one concept that we've explored, the idea of using these materials for drug delivery. The other concept is in biosensing. I mentioned earlier briefly that these materials undergo shifts in their optical properties as well, which we thought might be a useful mechanism for label-free biosensing if it were leveraged in these kinds of 3D networks. So if we change the polymer chain length that flanks these dynamic proteins and the total protein content in the gels, the thought is that we might be able to tailor the optical shift in response to Calmodulin ligands. So, as I mentioned earlier, there's this pronounced optical shift, and you see it most obviously here, and this is the result of the movie that was shown earlier. This internal portion is almost 100% translucent at the initial portion of the experiment, and then at the end of the experiment becomes quite opaque. And if we vary the molecular weight of the polyethylene glycol units that flank the protein and the total amount of protein that's present in solution during cross-linking, we can vary essentially the extent of this optical shift. So that's what's being shown here. From very little change in optical properties after addition of the ligand to quite substantial changes in optical properties, from say 80% of the initial optical transparency down to about 35% of the initial optical transparency. And importantly, these materials recover. So in each case, we get no significant shift we get no significant hysteresis, essentially. We don't get changes in optical properties that persist over an extended time frame. This is somewhat important because it suggests that the shift in optics doesn't have to do with protein crashing out of solution and forming what would amount to an inclusion body, but instead has to do with some sort of heterogeneity in the cross-links that leads to optical shifts. So this shift can be cycled as well. The volume shift, as I mentioned earlier, can be cycled several times. But importantly, this optical transparency shift can be cycled as well to a similar magnitude. And just as an initial demonstration of whether these materials might be useful in biosensing, we created 1536 well plates that contained hydrogels composed of the Calmodulin unit flanked by different molecular weights of the PEG unit. And in the case of the large molecular weight PEG units, which you may recall didn't undergo much of a shift in optics, we see some decrease in optical transparency, but it's not pronounced. Whereas in the presence of, the hydrogels that contain low molecular weight PEG flanking units, we see essentially these spots turn black. So this is promising and suggests that we may be able to use these kinds of materials for label-free biosensing. I should mention as a disclaimer at this point that right now we're sensing Calmodulin ligands, which is not what we're interested in sensing. So one of the significant directions we're heading in with these materials is broadening responsiveness so that we can respond, sense, and also deliver in response to more complex biomarkers. So what we think might be the advantages of these materials for biosensing ultimately is the specificity of the response, the ability to potentially multiplex, to respond to different ligands in the same hydrogel, the ability to adapt the structure and geometry of these materials, and then we may see significant increases in sensitivity and stability, but it's too early to tell whether or not these materials will be better than the gold standards. So just as a summary of this first piece, we can translate nanoscale conformational changes into changes in macroscopic material properties. These dynamic properties can be cycled and tuned. We can potentially use the physical properties of these gels in functional applications like biosensing and bioactuation. And then the hydrogel dynamics create a mechanism for triggered drug release at a particular time. And so this we're heading toward uses in both standard drug delivery applications as well as regenerative medicine. So I should mention there's a broad potential of these materials, and we're really exploring a lot of these different areas that are shown here. So one, there are hundreds of native protein motions that are well characterized, and we're using a series of them as candidates for new types of materials. And then it's also possible to engineer native proteins via de novo design, as well as via more complex phenomena like directed evolution, and we're exploring these kinds of concepts as well. The other piece that's important to note is that Henry Kopacek and others have done work with de novo order-disorder transitions in peptides that can be useful to form dynamic materials. So there's quite a lot of work going on in this area, and we're surely not the only group doing it, but we think there's a lot of potential to this work. So I'd like to shift gears a bit to another mechanism that we've been exploring now that is much more geared toward regulating stem cell behavior using a designed non-covalent interaction. And here, the concept involves a designed protein-mineral interaction. So as many in this audience would surely be familiar with, there's lots of work that's been done using materials to affect stem cell fate. And one of the common cell types that are used in this kind of an approach are human mesenchymal stem cells, which is the cell type that we're interested in as well. So for example, cell material adhesion, so basically cell interactions with adsorbed proteins, serum-derived proteins like fibronectin, vitronectin, this sort of thing, has been used as a mechanism to affect differentiation of MSCs, I'll call these MSCs for the rest of the talk, down the bone and fat lineage as well as the cartilage lineage. George Plopper at RPI is doing a lot of work in this area. Mechanotransduction, so the mechanical properties of the substrate that cells sit on is really vital in directing their differentiation potentially. And this was borne out in a Cell paper in 2006 that's really been quite famous since then. Really a significant piece of work from Dennis Discher's group. And Adam Engler, who did the work in Dennis Discher's group, is now doing some independent work in the area as well. Looking at the effect of the elastic substrate mechanics on differentiation of these cells down the bone lineage, the skeletal muscle lineage, and the neural lineage, and demonstrating the mechanics are quite important. And then Chris Chen's group and others have been working on spatially patterning these cells. And in a really elegant study recently, Chris Chen's group has geometrically confined MSCs on surfaces and demonstrated that the degree to which these cells are allowed to spread influences their ability to go down the adipocyte lineage and the osteoblast lineage. So in sum, these studies have really demonstrated that cell material interactions are vital for directing stem cell fate. What we've been interested in is sort of an alternative set of approaches where the material actually acts as a regulator of soluble signaling rather than as a regulator of sort of standard sort of typical cell material interactions. So what we're interested in is using materials to affect this interaction, the interaction between a soluble growth factor and a growth factor receptor on the cell surface. And there are a few different ways that one might envision doing this. So I want to go through and just introduce the different ways that it can be done, because we're actually exploring all of these ways, and then I'll focus in on data from really one example. So it's possible to engineer a biomaterial that can interact specifically with a particular growth factor, a soluble growth factor. So here the soluble growth factor is shown in red, the immobilized ligand shown in green, and then a cell adhesion ligand shown in blue. So in a system like this, a cell can attach to the surface, and that surface also regulates the availability of a soluble growth factor. And one can envision that as you change the material growth factor affinity, if the material is interacting with the growth factor active site, then there are possibilities of sort of regulating, up or down regulating growth factor activity by tailoring the material properties. So we're doing this with a series of growth factors. Another perhaps simpler concept is to immobilize inhibitors that bind to a growth factor receptor. So there are peptide-based and small molecule-based inhibitors that, when immobilized on a substrate, can be used to locally downregulate growth factor signaling. And this is potentially useful in a variety of sort of regenerative medicine applications. So here you would envision that as the material growth factor receptor affinity goes up, the effective growth factor receptor density on the surface goes down, and therefore signaling decreases. The concept that I want to talk about today is a different sort of approach where we're engineering the growth factor component, not the biomaterial component. So the concept here is that one can generate a molecule that has two functional units, a growth factor derived component that can signal to cell surface receptors and ultimately result in changes in gene expression, and a material binding tag that can bind to a particular type of biomaterial. So now you have a mechanism for essentially immobilizing a growth factor on a surface non-covalently. And this is the concept. As one increases the material engineered growth factor affinity or the density of engineered growth factor, you could envision increasing the effective growth factor concentration in the cell's local environment. So this is the concept that I'll talk a bit about. When we were initially developing this sort of approach, the concept was we wanted to develop components that were broadly applicable. So we started with a multipotent cell type that's been commonly used, that is human mesenchymal stem cells, or MSCs. We wanted a biologically active growth factor unit that promoted stem cell differentiation and could be readily modified. We wanted a biomaterial binding sequence that was specific and also could be bound to the material with variable affinity. And then we wanted to choose a biomaterial target well, one, we wanted an approach that could be applied to a broad range of biomaterials, but we wanted to choose sets of biomaterials that were clinically relevant. So this was the parameters we started with as we developed the approach. So the first embodiment of this is a molecule that contains a peptide derived from BMP2, or bone morphogenetic protein 2. This is a molecule that can promote stem cell differentiation down the bone lineage. There's an osteocalcin-derived, or actually osteocalcin-inspired mineral binding sequence that can bind to hydroxyapatite with strong affinity that can be potentially varied. And then there are hydroxyapatite surfaces. Hydroxyapatite makes up the major, by weight, about 75% component of native bone tissue, and also is ubiquitous throughout orthopedics. So it's a good sort of base material for us to start with because it's so common. So just a bit of an additional motivation for why we chose these components. So a bit of context for hydroxyapatite in orthopedics. So hydroxyapatite is now very commonly used as a coating on orthopedic implants to promote bone bonding to native bone in total joint applications, trauma, and dental applications. Bone void filler materials are used very often to try and promote bone formation in large defects as a result of trauma. It's also, as I mentioned, the major component of natural bone tissue, which means that it's also the major component of allograft, autograft, and xenograft tissues that are used very common in orthopedics. So one could argue that it's the most ubiquitous material that's used in orthopedic applications, and therefore is a kind of a good target for our molecules. The osteocalcin piece of this is really the sort of bio-inspired piece. So osteocalcin is a protein that binds with very high affinity to hydroxyapatite in nature. This is a structure of the protein bound to the surface of hydroxyapatite crystal. So this is from a Nature paper published in 2003. And you can see that there's this N-terminal helix that contains 3 GLA residues. GLA residues are carboxyglutamate residues. So they're not found too often in nature, but they have, basically they're glutamate residues that have an extra carboxylate group on them. So very highly charged, and therefore capable of interacting with calcium ions in the hydroxyapatite matrix. So that's what you're seeing here, is sort of geometric registration between the carboxyglutamate residues, the side chains on the carboxyglutamate residues, and the calcium ions that are present in the hydroxyapatite lattice. And here at higher magnification is what the sort of structure looks like. So our candidate sequences were from this N-terminal helix. The concept was to start with a sequence that was close to the native sequence, this sequence here, that contains 3 gamma E residues. Gamma E residues are carboxyglutamate residues. So GLA, gamma E, and carboxyglutamate all mean the same thing. Hopefully I'll try not to use them interchangeably. I'll just call them carboxyglutamate residues. And then if we then mutate these residues either to native glutamate, which contains 1 less carboxylate group, or to alanine, which contains no carboxylate groups, our hypothesis was that we could change the interaction between these peptides and the surface of hydroxyapatite. The other piece is bone morphogenetic protein 2, or BMP2. And a variety of previous studies have demonstrated that both in 2-dimensional and 3-dimensional culture, this is a molecule that can promote differentiation of human MSCs down the bone lineage. So a couple of references are shown here, but a lot of groups have shown this. And to varying extents, it can promote differentiation down the bone lineage. So this is what we were interested in, is essentially promoting osteogenic differentiation of MSCs using these kinds of engineered growth factors. So this is the library of molecules that we generated. The red sequence is derived from the knuckle epitope of BMP2. This was initially identified by Tanahara and co-workers in the 2003 paper, and retains a portion of the biological activity of BMP2, so it's capable of promoting bone differentiation. We have a polyalanine linker sequence, and then we have the osteocalcin-inspired sequences that I mentioned on the previous slide. And the concept here is that, again, these osteocalcin-inspired sequences can bind to the mineral surface, and the growth factor unit would remain active and capable of promoting stem cell differentiation. I want to define a term here that I'll use in a lot of the data that are in upcoming slides. EBGU3 is this sequence. So it's the sequence that contains all glutamate residues, no carboxyglutamate residues. And EBGA3 is the sequence that contains all carboxyglutamate residues. The important thing to remember here is that the EBGA3 sequence binds the hydroxyapatite and the EBGU3 sequence does not. And I'll describe the data that support that statement in an upcoming slide. So we started with hydroxyapatite particles, and question one was, can these molecules, these engineered growth factor molecules, bind to the surface of hydroxyapatite? So these are hydroxyapatite particles floating in solution in optical microscopy. So we started with fluorescently tagged growth factors and determined whether or not they could bind to the surface of these particles. So the EBGA3 sequence, the one with all 3 carboxyglutamate residues, binds very strongly. You can see these particles light up. So the fluorescently tagged molecule is there on the surface. If we look at all the rest of the sequences at the same level of exposure time in fluorescence microscopy, we can't see these particles light up. So there is quite a sequence dependence to this binding. The sequence that's most similar to the osteocalcin sequence binds much more strongly. And if we quantify it, it again sort of bears out the same data. The other sequences like the EBGA2 and EBGA1 sequence, so these are sequences that contain carboxyglutamate but lower number of residues, can bind to the surface, but with a significant decrease in affinity. And then, as I mentioned, the EBGU3 sequence that contains only glutamate doesn't really bind very strongly at all to the surface. There's very low levels of binding there. So these are the 2 sequences to be concerned with, EBGU3 and EBGA3. One important note here is if you look at the density of peptide on these surfaces and you compare it to native osteocalcin protein, the binding density of these peptides is about 6 times higher than native osteocalcin. So the interaction is really quite strong. If we look at sort of the mechanism for binding, and I won't get into this in too much detail, but just sort of briefly to give you an idea of how this might mimic what happens in osteocalcin binding. One, the binding is very fast. So within 5 minutes, we see 100% of the binding complete, essentially. And we've tried to run the experiment faster than this, and in each case, we see complete binding. So it's really quite a rapid immobilization. If we look at the circular dichroism spectrum of these molecules in the absence of hydroxyapatite particles or in the presence of hydroxyapatite particles, we see this kind of difference. So the EBGA3 peptide in solution, just in buffer, looks like a random coil. If we then add hydroxyapatite particles, you see development of peaks at 208 and 222 that are typically associated with alpha helix structure. So the suggestion here is that in the presence of hydroxyapatite, the binding between the peptide and the surface is stabilizing the presence of an alpha helix, and it might be interacting with hydroxyapatite in this sort of way. Random coil in solution, when bound, we see this helix forming. And importantly, this is what happens in osteocalcin. So we think we're mimicking essentially the structural transition that happens when osteocalcin binds to a hydroxyapatite surface. So getting back to the biologically relevant data. The next step was to immobilize these molecules on the surface of hydroxyapatite slabs and see whether or not they would promote stem cell differentiation down the bone lineage. So this is an electron micrograph of the surface of a hydroxyapatite slab. If we first look at the activity of these peptides in solution and we compare the EBGU3 peptide that doesn't bind to hydroxyapatite to the EBGA3 peptide that does bind to hydroxyapatite, but both contain the BMP2-derived sequence, what we see is that each can promote differentiation of MSCs down the bone lineage, here shown by alkaline phosphatase staining and alizarin red S staining for mineralized tissue. So in solution, these molecules are active, and if we quantify it, we see the same sort of phenomenon. We see activity of these molecules in solution. What we really cared about is whether or not these molecules were active when immobilized on a surface. So we took the hydroxyapatite slabs that I described in an earlier slide, bound peptides onto the surface, and then seeded onto those surfaces human MSCs in serum-containing medium. And what you see here is that the EBGU3 peptide does not promote increases in alkaline phosphatase activity or in alizarin red S staining on these surfaces, but that the EBGA3 peptide that binds to the surface of hydroxyapatite does result in significant increases in each of these markers. So both markers associated with bone differentiation. but both relatively non-specific markers associated with bone differentiation. So we looked in more detail at other markers that might indicate bone differentiation. So we looked at secretion of BMP2, production of BMP2, and production of osteocalcin in the medium, both markers associated with bone differentiation as well. And here we see that the EBGA3 peptide again increases the level of these markers. And one of the striking pieces of data is the osteocalcin expression level. So we see about a 10-fold increase in osteocalcin expression in cells that are cultured on the surface that's coated with this EBGA3 peptide. So when immobilized on the surface, these peptides appear to be active. What we were next interested in is whether or not this concept could be broadened both to different types of biologically active molecule and also to different types of biomaterial surfaces. If all we can do is immobilize a BMP2-derived sequence on the surface of hydroxyapatite, it's not as useful an approach as we hoped. So we looked at vascular endothelial growth factor. So this is a factor that induces differentiation of stem cells down the endothelial lineage, as shown in a variety of studies, and also is a mitogen for endothelial cells that promotes angiogenesis, which also has been demonstrated in a variety of previous studies. So this is part of the reason why we're interested in VEGF. So we took a VEGF mimetic sequence that was identified in a PNAS paper a few years ago, and we again linked that to our osteocalcin-inspired sequence, the one that contained the carboxyglutamate residues that bind to hydroxyapatite. Again, these peptides bind to the surface of hydroxyapatite with high affinity, and they bind quickly. So the data were roughly similar to what we saw with the BMP2 peptide. And then in the following biological data, I'll describe this peptide as VEGF mimic, and the other peptide as VEGF OCN. So what you see here is that when added in solution to cultures of human umbilical vein endothelial cells, or HUVECs, these are primary endothelial cells that are human. If we look at the cell number after a period of time in culture with these peptides, you see that the VEGF mimic peptide and the VEGF OCN peptide increase the number of cells in these conditions when compared to control, even when presented in solution down to the picomolar level. So this is consistent with the PNAS paper I described. These peptides are quite active and even at very low concentrations. But then importantly, when we immobilize these on the surface, when we incubate hydroxyapatite surfaces with either the VEGF mimic alone or the VEGF OCN conjugate peptide, the sort of modular peptide, what we see is that the VEGF mimic alone doesn't have an effect, and the VEGF OCN peptide increases the cell number. So what we're effectively seeing here is that these engineered molecules can also demonstrate a different type of biological activity, that is the biological activity of VEGF. Getting a bit short on time, so I'm not going to go into too much detail on this, but it's also possible to generate hydroxyapatite coatings on a variety of different polymer-based materials using biomimetic approaches. And this has been done by we and others in the field over a long period of time. The concept here is that the polymer surface provides functionality that leads to nucleation and growth of hydroxyapatite mineral layer. So our thought was if we could demonstrate that these molecules are active on the surface of hydroxyapatite coatings on biodegradable polymers, then it broadens the range of base materials for this approach. So in a paper that's just in press now at Active Biomaterials, we demonstrated this. So these molecules are active when immobilized on hydroxyapatite-coated biodegradable polymer surfaces. So I won't go into detail on the data, but happy to talk about this in more detail as needed or as interested. A couple of different clinical applications that become potentially exciting based on this technology. One is the possibility to spot materials with growth factor in specific locations. So you could envision implants where you might not want a bone-promoting growth factor or a vascular-promoting growth factor everywhere. You might want it only in specific locations. And we can do that here with this, the engineered BMP2 peptide. And as we wash the surface, the peptide doesn't come off, so the affinity is quite high. The other thing we can do is paint materials with engineered VEGF. This is a case where we essentially take a needle that's used in the operating room, we dip it in a solution of fluorescently labeled modular VEGF, and then we write on the surface, in this case UW, and you can see that it doesn't wash off again. We can also paint sutures with multiple growth factors and put the growth factors in specific locations. It's hard to see here, but essentially this looks like a candy cane. There's a green labeled growth factor here and a red labeled growth factor here along the length of the suture. So it's possible then to use sutures as a mechanism to spatially control growth factor delivery, and we're looking into these sorts of materials for bone tendon interfaces and that sort of thing. This is just another picture. The postdoc who did this work wants me to show every drawing that they do. They have an artistic bend, so if I don't show this, I get in trouble. But you can draw a VEGF as well on the surface. So as I mentioned earlier, we think there are clinical applications for some of this work because of the ubiquity of hydroxyapatite in vivo. And we've explored this recently on hydroxyapatite-coated bioresorbable bone screws. So in this case, we have a sheep model of bone tendon healing. We create a tunnel in the bone, in the tibia of these sheep. And then we place a graft, and we use an interference screw to stabilize that tendon graft inside the defect. And then over time, we look at whether or not the molecules that are essentially present on the surface of these screws are affecting healing. And what you can see is in the presence of a control screw, you see the tendon, and then you see the native bone, but you don't see a lot of new bone formation, and you also don't see a large abundance of sort of progenitor cells at the interface that are getting activated. In the case of the screw that's releasing, or that contains the molecule that we've engineered, you see new bone formation extending away from native bone, and then it's not shown here, but if you look at higher magnification, you can see mesenchymal cells at the interface sort of getting activated. So we haven't looked at in a lot of specificity at what types of cells these are, but it's pretty clear that there's a different level of cellularity, and it appears that they're sort of mesenchymal precursors at that interface. So the histology scores are much higher in the case of the coated screws. This is a paper that's just coming out in arthroscopy. So just to summarize this last piece, osteocalcin hydroxyapatite binding can be mimicked using some of these synthetic peptide libraries. We can immobilize then biologically active molecules like engineered BMP2 or VEGF. The rapid binding potentially enhances clinical applicability, and then the rapid stable binding allows for spatial control over biological activity, which maybe opens up new avenues. And I'll end with this rather creepy smiley face that was drawn by the same postdoc. So lots of directions that we're heading in with these sorts of interactions, and I've had some great interactions with students and faculty already during the visit, and happy to talk more about some of the other work we're doing in this area. Let me just briefly acknowledge the people who did the work here, really a dedicated group of students and postdocs and technicians in the group. Jae-Sam Lee and Jae-Sung Lee have done the work with the engineered growth factors, and you can imagine this gets very confusing when we're drawing out lists of authors and that sort of thing. Juji Sui and Bill King have really done all the work with the engineered proteins that are dynamic, and Bill is really heading in the direction of using these to trigger changes in vascular growth in three-dimensional networks, so we think there's some exciting work down the line there. And Jim Melenda is a technician in the lab who's been involved really in both of the projects I described. I also want to acknowledge sources of funding for the work, and would be quite happy to take any questions that you might have.